at this webinar. To motivate my project, I'd like to show you these beautiful pictures of HeLa cells and of hippocampus, and kind of remind you that the microscopy and fluorescence imaging can provide you both quantitative and qualitative information inside the cells. And on the left, what you see is HeLa cells undergoing apoptosis. And then on the right are neurons. It's from Rainbow, where each neuron is colored differently in this brain section. And I would like to show you one other image, which is an image of a tissue section from a mouse E16.5 fetal liver, where green shows you the nucleus, and then the magenta shows you the cell membrane. And what I'd like to highlight is that in this tissue section alone, you can see that how different each nuclei look in terms of size, shape, and morphology. And currently, we don't really understand why we see this heterogeneity, and if this kind of like the variation in size and shape matters at all in the context of development. And kind of on the other side, which is in disease, kind of like the visual or image-based phenotypes have been used to assess the impact of some of the mutations in clinically relevant genes. I'm going to show you two examples. One example is the mislocalization of FUS protein to the cytoplasm in some of the ALS patients, where normally FUS protein resides in the nucleus, but in cells of some ALS patients, the FUS protein kind of mislocalizes into the cytoplasm, where when we look at the nucleus to cytoplasm ratio, they're lower in the ALS patients. And the other example is the pathogenic line mutations, where some mutations alter the nuclear morphology. In the slide, I have examples of progeria cells. So when you compare the lamin B1 staining and also the nucleolus of the wild type and the progeria cells, you can see that in progeria, there are some aberrations in the nuclear morphology, and also the nucleolus is enlarged in some of the progeria patients. And the changes in the nuclear structures can be seen during cell type differentiation as well. In this review paper, they took a look at some of the nuclear markers in the embryonic stem cells and neuroprogenitor cells. And what you can see is that some of the markers, such as heterochromatin protein and nucleophosmin and lamin B, kind of like patterns in nucleus, are quite different between the stem cells and neuroprogenitor cells. And kind of when you look at the histone modifications as well, you can see that in different cell types, they have kind of different patterns of chromatin architecture, which is highlighted in the right paper. So recently, there has been a big advances in single cell genomic technologies, and in our department as well, we do a lot of single cell RNA sequencing and single cell attack sequencing of in vitro ES embryonic stem cells and also the in vivo mouse models. So kind of like one of the questions we would like to answer is there a relationship between the nuclear features that we see from microscopy and the single cell molecular profiles we get from single cell genomic methods? So one of the questions we would like to answer is can we explain some of the transcriptional heterogeneity we see in our single cell RNA-seq data with differences in nuclear morphology in our images. So as you can imagine, if you were to do a single cell RNA-seq on embryonic stem cells and your progenitor cells, you'll see that the gene expression patterns are widely variable. But if you were to have some sort of image-based phenotypes along with the gene expression profiles, you can imagine some genes can be kind of further divided into different morphological bins, and that the morphological information can explain some of the variation we see in gene expression. So to get at this question and to correlate gene expression with nuclear features, we're using a method called visual cell sorting, which is a high throughput image-based method that allows us to separate cells based on image-based phenotypes. So for the bulk of this talk is to kind of show you how this method works and update you on some of the improvements that we made for this protocol. So first, we would have to get the images of the cells of interest. And in the published protocol, one of the requirements was to use or have the cells express this photoconvertible protein called dendrin-2 that is normally green in its native state, but upon UV activation, it photoconverts to red. And we use the system to label cells with the visual phenotype of interest. So how this method works is that when we have our images and when we do some sort of image analysis afterwards, so in this example here, we have cells that are flat and then cells, round, and some cells that are star-like. And what you can do is you can kind of use the UV activation to encode the phenotype. And this can be, this is great in that we can get multiple phenotypic bins by tuning the time of illumination. So, and in our paper, what we showed is that we can illuminate these cells for 50 milliseconds, 200 milliseconds, 800 milliseconds, and we can get 3 activated populations along with the non-activated populations. And the selective activation is done by the use of a digital microarray device, where digital microarray device is an array of a bunch of mirrors that can either turn on and off, so that sometimes you can get a UV light go through in some regions and not. So you can kind of control or when and where the cells are activated in the field of view to allow for selective activation. So once you do that for all field of view, you can use first and activated cell sorting to physically recover the cells that you have activated. And one of the examples that we have in the paper, one of the applications is that we have used visual cell sorting on RP cells treated with paclitaxel. So when you treat the RP cells with Taxol, some cells kind of maintain its normal shape, but some cells exhibit these lobulated phenotype. So in the paper, Nick kind of sorted normal and lobulated cells and did single cell RNA-seq on them. And if you're interested, you can check out the paper. So some of the key advantages of visual cell sorting is that you can physically recover 4 visually distinct populations. And the method is pretty fast, so it's high throughput. And depending on what objective you use and what kind of image analysis you do for doing the activation. But on the faster side, we can phenotype around 2 to 3 million cells per day. And all the hardware and software is from Leica, so it uses commercially available reagents. So if other people want to use it, you can use it. And they're also compatible with single-cell genomic assays after sorting. But the original protocol kind of had its limitations as well. The one big limitation is that it was only applicable to live cells, and therefore phenotyping was limited to live cell dyes and frozen protein tagging. And therefore, you kind of need to have the cells to express dendro2. So if your cells of interest are from a primary sample, or if they are really hard to transfect, this was a huge limitation. And because of the turnover of the dendro2 protein, the experiments were only limited to 12 hours. So what we did in the past year or so, we've been kind of optimizing and improving the protocol by making it compatible with cell fixation. So I'm just going to show you some of the results that we got and kind of convince you that we can do this with fixed cells. So I'm showing you here is kind of like the pilot experiment where we co-cultured green and red cells together, and then we fixed them on the plate, and then we dissociate by scraping, and then we fact sorted green and red cells. And the point of this experiment was to kind of demonstrate that you can indeed dissociate fixed cells and sort them, and get single cells or single nuclei afterwards. So when we sorted them in green and red differently, what you can see is that in the sorted 50 nuclei, you only see green cells, and the sorted red populations, you only see red cells, and you don't see any capture of red cells in the green population and green cells in the red population. So this kind of confirmed to us that when you do these experiments in fixed cell conditions, you're not getting that much of doublets. And cell fixation is really nice because it allows us to use photoactivatable dyes instead of genetically engineering cells to express dendro2. So when you use a dye that has similar properties to dendro2, that can be photoactivated with UV light. And when you can conjugate this dye to an antibody, along with some control green dyes, and then when you do randomly activate for different amounts of time, what you can see is that you can recover up to 4 different populations at the flow sorter. So this kind of told us that we can indeed use this antibody system to do some visual phenotyping. And also, what we saw is that when we do some sort of these experiments and then sort nuclei, we can apply single-cell TAC-seq and single-cell RNA-seq on them. So what I show you here is that a test experiment that has a mixture of human and mouse cells together. And when we follow the same protocol where we fix the cells on the plate and then detach them and sort them, and then when we do TAC-seq on them, you can see that we mostly get singlet populations as well as some doublets. And this is also and then this is also true with the single-cell RNA-seq method that we have, or SkySeq. And one thing you might be wondering is that does the transcriptomes look comparable with the standard data? So what we did was we took a mouse embryo and then we isolated nuclei from them. And then we did like a test, which was also an experiment where we plated these nuclei and then we imaged for multiple days and then sorted them afterward and performed sRNA-seq. And this is what the UMAP projections of this data look like, where the red is the reference data, and then the blue is the data from this mock experiment. And as you can see is that without any alignment or anything, the data from these nuclei overlap really well with the reference data. And when we use the cell type annotations from the reference data, you can see that we recover most of the cell types that are in the embryo. And to really make sure that we're indeed getting intact nuclei, we re-image the sorted nuclei and kind of confirm that they are indeed intact and they're mostly singlet populations. Okay, so with kind of visual soul sorting compatible with self-fixation, what we ultimately want to do is to apply this method to nuclei derived from mouse models, either the in vitro embryo stem cell model or the nuclei from the in vivo mouse embryos that are in the later stage, so around E11 to E15. And the proposed pipeline is to isolate nuclei from either of these two samples, and then we immunostain for different nuclear markers, and then based on what we see in the images, we can sort them with visual soul sorting. So we would get some populations that look one way and some populations the other way. And then we can sequence with a bunch of different techniques, such as gene expression, chromatin stability, and chromatin interactions. Yeah. And one marker that we are currently using to do visual soul sorting on is the Nucleolin marker. And what we see in literature and in ourselves is that the size and the number of nucleoli are highly heterogeneous within a cell population. So this is a picture of neuroprogenitor cells stained for nucleolin in red. And what you can see is that even within a population of same neuroprogenitor cells, you can see a wide distribution of different sizes of nucleoli and also the number. And in fact, the size of nucleoli, they kind of decrease during ESC to MPC differentiation. So we took images of embryo stem cells and neuroprogenitor cells and some of the intermediate time points along the way. And we stained for nucleolin. And when we compute the area of nucleoli inside the nucleus, what we observed is that they go down over this this differentiation pathway. So what we want to do is kind of apply visual soul sorting and sort based on either the nuclear size or number. And we first want to do this in the mouse embryo because of there's so many cell types in the mouse embryos that that when we kind of bin them into different sizes or number, that we are curious to see if there's some sort of correlation or differences between the cell types. So so far we've been working on getting the method to compatible with mouse embryo nuclei, and also we're making this as fast as possible. So in this coming months, we're planning on applying to soil sorting to sort based on the nuclear area, and then apply different single cell genomic methods such as RNA-seq, SkyTaxic, and Hi-C, and hopefully to map kind of like the relationship between morphology and some molecular profiles. And we are not only limited to using a nucleolin marker. So one other marker that we're thinking of using is heterochromatin alpha. We can also use a DNA dye as well to visualize these little heterochromatin foci and ask if the amount of heterochromatin that we see in the images correlate with chromatin accessibility, for example. So we can kind of explore all the markers we can use and also the methods that we can use to profile them. So kind of to conclude my talk, I've kind of told you that the nuclear structures, they undergo huge changes during cell differentiation. And I told you about a visual cell sorting method that we had developed in the Faro lab that allows us to separate cells based on visual phenotypes. And hopefully when we apply this method to nuclei derived from mouse models, we can characterize some of the nuclear features that we see in the context of mouse development. So with that, I'd like to thank the Fowler and the Troutman lab, and also Doug, Cole, Bill, Jinshin, Christine, Shiram, and Eva, and Gong.